Thank you. And thanks everyone for getting up so early. So why does structography still get things wrong? We have these brand new scanners with ultra high gradients. We have highly accelerated sequences, so we're able to collect data at much higher resolutions, higher B values, higher SNR than ever before. So we can get very accurate estimates of diffusion orientations. However, our spatial resolution is still somewhere in the scale of about 1.5 millimeters at best in vivo. And this means that for many of those voxels, there will be more than one axon population in there. And as soon as this happens, there will be multiple configurations of these axon populations inside a voxel that give rise to a very similar diffusion profile. And this means that when tractography reaches that voxel, it will not know which of these peaks to choose, and so it will have to make an arbitrary decision. And sometimes these decisions will be correct, and sometimes they will be incorrect. This is an example where tractography took an obviously incorrect decision and turned from one bundle from the corticospinal tract onto the corpus callosum. Now, this is a situation where you can tell with very little knowledge of neuroanatomy that we took a wrong decision, took a wrong turn somewhere, but in most cases, something that we see in tractography results, it's hard to tell if it's actually true or not, and that's why we still need ground truth on connectional anatomy to inform diffusion MRI tractography. The challenge with this is that this ground truth typically comes from modalities that are collected at very different spatial scales than in vivo human diffusion MRI, may even be collected in different species, so it's difficult to integrate it into human diffusion MRI tractography. However, it is important to try to tackle these challenges because doing so would allow us to establish optimal diffusion MRI acquisition and analysis techniques by having an objective criterion for accuracy, and it would allow us to identify realistic failure modes as opposed to the cartoon kissing and crossing fibers that I showed you in the previous slide. And having these realistic failure modes would give us ideas about how we can improve our current tractography methods. And last but not least, it would allow us to build accurate models of connectional anatomy from the ex vivo data that this ground truth typically comes from, that we can then use to improve analysis of in vivo data, which is what most people in here are interested in. And I'm going to show some preliminary results for how we're tackling each of these points. So the two sources of ground truth that we're using for connectional anatomy, one is chemical tracing in macaques. This allows us to visualize end-to-end trajectories of axons from an injection site to all the terminals, and we can compare that to tractography from the injection site. However, it gives us access to only a single fiber population at each voxel. We see the axons from the injection site, but not everything that's crossing with it. On the other hand, using optical imaging in human samples, we can get all the fiber populations inside one voxel, but the techniques that we're using at the moment to do that have some technical limitations, and so they do not allow us to follow bundles all the way from one end of the brain to another. We can only do this in small samples at the moment. So these two sources have complementary strengths and weaknesses. First, the tracing studies. So work that has been done using tracers in macaques has shown that the large white matter pathways, like the corpus callosum, the cingulum bundle, the uncinate, etc., are not monolithic structures, but instead they're made up of many smaller axon bundles that use these large pathways as highways, and they merge on and off the highway at different points along the trajectory of those highways. And work by Suzanne Haber's lab, where they have been doing these tracer injections for many decades now, has shown that there are clear topographies of these smaller bundles within these large pathways. So this is an example of topography going, the injection sites going from more dorsal to more ventral, so green to yellow to red in the cortex. And she has shown, her lab has shown that that same topography is preserved inside the internal capsule. And we are able to replicate this green to yellow to red topography using diffusion MRI tractography in the same monkeys. This is in the middle column. And then on the right, using the B equals 10,000 human in vivo data from the MGH Human Connectome project. So this suggests that ideas that we can get by comparing the diffusion to the tracing in the monkeys, we can then apply to improve how we analyze the human data. So in these monkeys where we have both tracing and diffusion, we are able to label the axons under the microscope, assemble all this data across all the slides to create a 3D model of the tracer injection. And then we have 4.7 Tesla whole brain ex vivo diffusion scans of those same monkeys. So we can run tractography from the injection site and compare voxel by voxel to the tracing. Some preliminary analysis where we have compared different diffusion sampling schemes, deterministic versus probabilistic tractography, as well as different both model-free and model-based approaches to reconstructing diffusion orientations, show us that the top performer at this moment seems to be a model-free approach combined with probabilistic tractography. And another interesting result is that we do not see a huge benefit from using multiple B-values versus just a single B-value. Another kind of question that we can ask of this data, because we have scanned a few different monkeys, is does it really matter if we have the tracing and the diffusion in the same animal? So if, let's say, we compare the accuracy of probabilistic versus deterministic tractography, just to give an example, if we compare it on the basis of tracing and tractography in the same macaque versus tracing and tractography from tracing and diffusion data from two different macaques versus tracing in one macaque and diffusion in a set of different macaques, do we get the same answer about which method is better? And what we see in our preliminary results is that while the big trends tend to generalize, some subtler ones do get diluted. And we do see, even though there's lower variability in macaques than in humans, we do still see higher variability across animals than within animals, suggesting that it is important to have the tracing and the diffusion in the same brain. And because we're not going to be able to compare all possible methods in this data, we are organizing an open tractography challenge. The website shown right here, irontrack.mgh.harvard.edu. You can go and sign up and download the training case already. And the first results will be announced at MICAI this October. One special aspect of this data is, as I said, that we have the tracer injections and the diffusion in the same animals. Another thing is that we have quite a flexible diffusion MRI sampling scheme. So you are able to use methods that require single shell, multi-shell, or even DSI data. We are going to have an overall winner where contestants are going to be able to choose the sampling scheme that best suits their processing approach, as well as an HCP winner where contestants will compete on the specific two-shell acquisition scheme that's used by the Lifespan and Disease HCP. So I encourage you to participate and let me know if you have any questions about the challenge. As I said, looking at the tracing versus the tractography in these macaques gives us ideas about what are some of the failure modes, some of the tricky areas that tractography gets wrong. And those are particularly switching areas where those small axon bundles jump from one highway onto another. And so these give us ideas about what particular areas of white matter we should follow up on in the human data, where we cannot, as I said, at the moment, do a whole brain imaging. So the approach that we're using is polarization-sensitive optical coherence tomography, or PSOCT. It relies on birefringence, which is an optical property of anisotropic tissues, and in the brain, and specifically in myelin, the birefringence is due to regular pattern of arrays of lipids and proteins. So PSOCT gives us a direct measurement of the optic axis of the tissue, so the fiber orientation in the plane orthogonal to the incident light, as well as a map that's called retardance, and is more or less dependent on how much myelin is there, and we can use the retardance map to register to diffusion or MRI data in general. A couple of advantages of this approach is it does not require any image processing to extract the fiber orientation, so that color map comes out as a direct measurement of the scan. And also, it images the superficial layer of tissue before cutting it. And that means that we do not have nonlinear histological type distortions from slice to slice. The disadvantage is that at the moment, we can only do it in small samples. So it helps to know on the basis of the tracing studies, what specific parts of white matter have those tricky areas so that we focus there. So this is an example where we found one of those tricky switching areas using whole hemisphere ex vivo diffusion data from human hemisphere. We cut out the area of interest and did further diffusion MRI scanning at high resolutions, 250 microns, in a small bore 9.4 Tesla scanner. And by finding that area with the most complex and interesting fiber configurations from the high resolution diffusion data, we focused the PSOCT scan in that region. You can appreciate from this movie how well aligned the slices are to each other. So this is how the data comes out of the scan. We do not have to do any registration from slice to slice. In some preliminary results, just an example of the kinds of things that we can learn from this data. So we compare the PSOCT on the left to diffusion orientation estimates, starting at the top row from the native resolution of 250 microns of the diffusion data, and downsampling down to 0.5 and 1 and 2 millimeters. And the different columns are different approaches to reconstructing the diffusion orientations. Across the board, we see that things are doing okay, even for the simpler models, up to a 1 millimeter resolution. And then things really break down after that. And this is a suggestion so that for in vivo resolutions in the future, it is worth trying to target 1 millimeter or even sub-millimeter spatial resolution. And I'm going to show an example to finish on how we can, we're planning to use this data to improve how we analyze in vivo data. So I'm going to show a case study focusing on the superior longitudinal fasciculus. And what you see here on the left is a nice delineation of the three components of the SLF using the B equals 10,000 MGH human connectome data versus the SLF that we would get from the kinds of data that we were able to collect 10 years ago, where we see mostly the SLF3. So the question is whether we can train algorithms on the nice data on the left to reconstruct the SLF much better in the kinds of routine quality data like the one that you see on the right. And Chiara Maffei recently showed that indeed you can feed the kinds of models of the SLF that we get from the high quality B equals 10,000 data into the anatomical priors that we use for trachea, and use trachea to reconstruct highly accurate, do a highly accurate reconstruction of all three components of the SLF using just B equals 1,000 60 direction data. So we can do kind of a quality transfer from higher quality data to more routine quality data that can be acquired even clinically. So the idea is in the future, as we improve further the models of connectional anatomy that we have using the ex vivo data, we can feed it into this framework and improve our in vivo reconstructions. So focusing on the SLF, there are certain things that even in the highest quality structography data, we cannot be sure of. And one such question is, how rostrally does the SLF1 terminate? There are a lot of contradicting results from the literature. Sometimes it ends near the precentral area. In other cases, it goes further anterior. And some gross dissection studies suggest that indeed it should go further anterior, but potentially with structography, it ends prematurely because of other crossing fiber tracts. So we focus on that area of uncertainty, the potential anterior terminations of the SLF1 in a human postmortem frontal lobe. We got two slabs that contain that area and focused on the part of those slabs under the superior frontal gyrus. So, oh, this wasn't right. Okay, so this shows the whole hemi tractography from this hemisphere. The blue is the SLF1, and it shows that it actually terminates near precentral area, and the orange is the cingulum shown just for context. So now you're starting to see the blocks that we acquired in the 9.4 Tesla scanner and the diffusion orientations from those higher resolution 250 micron scans, showing that indeed there are those green fibers that may be those more anterior terminations of the SLF, but there are also those blue fibers that are the ones that are potentially causing tractography in the lower res data to terminate prematurely. Looking at the PSOCT data, we confirm this, and we see that those little green bundles, so these are, the color scheme is slightly different, so these are the ones that are going inferior to superior here, are what are potentially causing those premature terminations of the tractography. So we hope to use this high-quality data, ex vivo data, to incorporate it into algorithms that can then improve reconstruction in vivo. And I would like to end by thanking Robert and Chiara, who are doing a lot of that postmortem work, as well as all my collaborators. And last but not least, we are hiring, so we're looking for postdocs for our Connectome 2.0 project, which will build their next generation Connectome scanners. So we're looking for people in the whole range from acquisition to analysis. And if you're interested, please talk to me after this session. Thank you.